Welcome to Race and Medicine, folks. We'll just give people another minute to get in from the waiting room. Okay, let's go ahead and get started. Uh, Sorry about that. This is Race and Medicine. If you're here for the first time, welcome. If not, you know what's going on. Sorry, I keep. This is a course as well as a speaker series, so we have some people here who are from the Stanford community and others who are students. We this series, which began in the fall, this is now our 22nd event this year. Uh, we're co-sponsored by all these different units at Stanford who contributed money and time and energy to put the series together. So we're deeply grateful to all of them. We always begin with a land acknowledgement to the Muwekma Ohlone tribe, since we are on their ancestral land, which is of great importance to the Ohlone people. So, because of our values of community and diversity, we have a responsibility to acknowledge that and to honor and make visible the university's relationship to Native peoples. The land acknowledgement will also be in the chat, along with a link to the Ohlone tribe, and you can check in with them if you want to learn more. Next time, sorry about that. Next time, we have, for the third time this year at Stanford, Professor Keith Walu of Princeton University, who this time will be talking to us about pain. Pain with a psychogenic overlay, gender, race, and the political ethics of pain relief in U.S. society. And now I'll turn it over to Jasmine to introduce our guest for today.
Welcome, everyone. I am so excited to be able to introduce our speaker today. We are joined by Dr. Alice B. Popejoy, who is a computational biologist with a PhD in public health, genetics, and a certificate in statistical genetics from the University of Washington. Dr. Popejoy's research is situated at the intersections of computational genomics, human population genetics, clinical genetics, and bioethics in the Department of Biomedical Data Science and the Center for Biomedical Ethics at the Stanford University School of Medicine. She's also involved in several NIH-funded consortia, including the Clinical Genome Resource, or ClinGen, and Clinical Sequencing Evidence Generating Research, or CSER. And she's the co-chair of the Ancestry and Diversity Working Group and chair of the Data Access, Protection, and Confidentiality Working Group. We are so excited to have her share more about her research, so please join me in welcoming Dr. Alice Popejoy.
Oh,
Alice,
there. Can you hear me now? Wonderful. Thank you so much for having me, Jasmine and Paul, for inviting me, and for everyone who's interested in participating today. I hope you will participate also. We have some interactive elements of today's presentation, so I won't just be talking at you the whole time. And actually, I think Jasmine's going to put in the chat a link to Poll Everywhere. So I will be using Poll Everywhere. You can either log in through the link, or you can actually use your phone to text message to access the session, and then just add your responses kind of as we go to a couple of these, just as conversation starters. So I'm not going to be calling on anyone, putting you on the spot, to forcing you to say anything in front of the big group. But if you could participate in your own way at your own time in the way that works for you, that would be really wonderful. So the title of my talk today is What is a Population? Investigating Race, Ethnicity, and Ancestry in Genomics Research and Clinical Genetics. And this is a really big question. What is a population? I think a lot of us have some preconceived notions about populations. Different folks in different disciplines might be thinking about it differently. So just to get a sense of kind of where we're all starting on this question, get a baseline understanding, I'd love if you could to log into Poll Everywhere. Looks like some people are already doing this, fantastic. Texting to the Alice Popejoy 558 22333 in order to join the session, and just type whatever word comes to mind when you think of a population. Give folks about another minute, 30 seconds maybe or a minute to do this. So people is a big winner, group or groups, characteristics, number, diversity, demographic, communities, culture, others. Wow, awesome. I really like how it's a group of people, right? If that's what we're seeing from your responses here, that you think about generally population is a group of people. And I think that's a really actually wonderful way to think about it, because you can group people in any number of ways, right? And we group people in different ways for different reasons, and in different contexts. And in the United States, this has been a particularly fraught issue over the decades. Obviously, diversity, equity, and inclusion have become a big part of the national, international conversation, and more people in genetics and genomics are paying attention to these issues. But there has been literature over many decades kind of debating how we group people into populations. So the idea of racial classification in particular in research and medicine has been hotly debated, and we still have scholars who are experts in their disciplines on opposite ends of the ideological spectrum with regards to how something like race or racial identity can and should be used in research and medicine. So I'm going to be talking about sort of the clinical genetics side of this and some of the research we're doing there, but also about how this affects basic genomics research in the biomedical context. And the reason for that is that there is a sort of a feedback loop in the research to precision medicine pipeline, such that the work that we do as basic genomics researchers feeds into genetic testing, interpretation, and even diagnoses. And then that evidence is curated by a series of different resources and volunteer curators. And then the information that we have, that's sort of the genomic knowledge base, if you will, feeds back into basic research in terms of helping us prioritize what to study, which populations to study, and sort of the trends that we're seeing from the clinical setting. So it's really important that as we work towards solutions for figuring out what a population is or how to appropriately group people into quote unquote populations for different purposes, that we're thinking about this interconnectedness between research and clinical care, and not just pretending like we can solve one over here and the other over here and it's going to magically work between the two. So I've been spending some time thinking about what are the different limitations with regards to diversity in research and medicine all along this pipeline. So the first is probably the most substantive issue we face is the identification of genetic risk factors, right? What do you test for in clinical genetic testing? Well, what we know about is based on the genomics research that's been done. And we know there's a lack of diversity in genetics. Also, results often come back as sort of population specific, like this is what we know about this group of people, but we're not sure whether this tracks in other places. And also there are really inconsistent descriptions of populations. So I'll give you some examples of that. So you may have seen this slide. If so, that's wonderful. It was a result of some work I did as a PhD candidate at the University of Washington in public health genetics with Malia Fullerton. I'll tell you just briefly background on this. I was really frustrated as a graduate student seeing this pie chart on the left everywhere with all of our guest lectures. It's 96% of genome-wide association studies are done in people of European ancestry. And often it was like an asterisk on someone's presentation about their research. Like, just take it with a grain of salt because this might not replicate in other populations. And I'd always be like, are we good with that? Everyone's cool with that? We're just gonna say this might not port across populations, but like, what's anyone doing about this, right? After several years of seeing the same slide, I was like, these are old data, and are we making any progress? Because it seems like everyone's aware of the problem, but what's being done to solve it, right? So the simplest thing to do, I felt, was just to go in and look at the GWAS catalog and see how much progress had been made. And initially, at first glance, you see this 81% people included in genome-wide association studies are of European ancestry. So there's some improvement, but the majority of this improvement here in diversity and inclusion is comes from studies that are conducted in Asian countries by Asian researchers on their own population. So this isn't necessarily data that's being generated and studied in the US and Europe, where the majority of GWAS are being conducted. This is genomics taking hold in other parts of the world, which is amazing, and that's wonderful for those countries. But Nature is a British journal, and I'm based in the US, as are most people doing GWAS, and thus I didn't feel like Europe and the US could really take credit for that growth and diversity. I think this is a more honest pie chart, so excluding those sort of Asian specific studies. That's really not a whole lot of progress, and it's really a problem. I know there have been some diversity efforts in genomics since 2016, but I think we still have a long way to go in terms of actually just knowing about what's the genetic variation that's leading to disease across populations. We're starting at baseline pretty much. The other issue I mentioned is that population descriptions are all over the place. It was not trivial to do this analysis because the way that people are described in populations is all over the place, right? You have things like Hispanic cases. That's ethno-linguistic. So whether someone speaks Spanish or not, or they're from a Hispanic culture, but people in Latin America don't identify as Hispanic. So what does that mean, right? Black cases or controls, like that's a racialized category. What information is that really giving me about who this result applies to? Because it's such a broad racialized conception. And then of course you have all this granularity on the European side with very specific villages and things like that. So it's all over the place in terms of how GWAS are described. They've actually now implemented Ancestro, human ancestry ontology, for the GWAS catalog. So they're making progress in creating ontologies and structures, but the issue is the implementation and whether people are actually going to adopt that ontology and whether that works for diverse populations, not just how we conceive of them in the US and the UK. So that'll be interesting to see. So I was working on this analysis and this issue on lack of diversity in genetics, as I mentioned, as a graduate student. I also had a lot of other sort of computational projects and methods development projects, but I came down to Stanford to talk with Carlos Bustamante about his original paper that used to get cited in all the conferences, and now my pie chart sort of replaced his in some cases in advance to just touch base and see if he had any advice for me. And he actually offered to interview me for a postdoc position and continue this work more formally, working with him on the clinical genome resource, which is an NIH-funded resource called ClinGen. And I started with Carlos in 2017, the Ancestry and Diversity Working Group for ClinGen. So for the last 3.5 years or so, we've been working on this question of how race, ethnicity, and ancestry should really be used in clinical genetics. And of course, sort of having this understanding that that's related to research as well. So first and foremost, how are these measures defined? How are they used in research in medicine? And then how should they be used, right? Because we can't just come out with guidelines without understanding what's current practice and what needs to be considered. So these are basic, foundational research questions in this space that we've been exploring. And the first thing I figured out pretty quickly is that there are no standard definitions of these diversity measures, which causes a lot of confusion. So there's a lot of social scientists who have done lots of work around this, and I'm heavily relying on that work on critical race theory and other sort of social science findings. But when I talk about race, I'm going to provide just some useful definitions for you so that in my presentation, what I mean when I say race is that this is a socio-political mechanism for classifying individuals. It's often tied to status and power, and it reifies an ideology of inequality. So it's fundamentally, you can't have race without power, right? Ethnicity is often used interchangeably. You see things like race/ethnicity, but they're not the same thing. Ethnicity often refers to something more cultural in nature, linked to community, religion, language, even cuisines and traditions. Whereas ancestry, lots of different cultures have different meanings of the word ancestry. For some, it might mean traditions. It might mean something that they inherited from their families that's intangible or cultural in nature. But when biologists talk about ancestry, we typically mean that you literally inherit chunks of DNA from your ancestors through your parents and through their parents' parents, et cetera. And you can actually trace those lineages back using genotype data and comparing them to reference panels to sort of determine how much ancestry on average you share with other people in different parts of the world. So these are some useful definitions, but the problem is the way we actually implement categorization of people into groups in the US is not clearly delineated among these different meanings of those terms. It's all mixed together and it's fluid. So although you have these separate circles for race, ethnicity, ancestry, terms like Asian that fall in the middle could be used in any which way. You might say something stereotypically about my Asian friend so-and-so, right? That's a racial stereotype, or it's a racial identification of somebody based on some identity you've either assigned them, or maybe they self-identify as Asian, but it's sort of a racialized thing to say, right? Versus like, last night my family had Pan-Asian food. Okay, that's an ethnic reference sort of to this type of cuisine. Or you might be a researcher and say, these are my study participants of Asian descent or Asian ancestry. And that's sort of a nod to some sort of biogeographic link to a country that's in Asia based on their DNA or their genetic background. But more than half the world's population lives in Asia. So how useful is that really? Is it inherently useful for research in medicine? Or is it useful insofar as that it reifies some racist notion we already had about being Asian as some meaningful category because of sort of the race and ethnicity component? So it's really important to think about the labels and the terms that we use and why and when and how we use them, and whether or not that's truly scientific and precise, or whether it's just sort of by virtue of being in a society that's inherently founded on the need to separate people on the basis of some arbitrary physical characteristics as a means of power and control and basically oppression, essentially. So important to think about that. I want to note about ancestry. I do love this tree of life. Oops, I already showed you the answer, but basically in the grand tree of life that includes mostly bacteria here, in terms of genetic diversity, it's like a big family tree of genetic sequences. Humans live on this tiny little branch called a pistoconta with trees and fungi. So if you think about ancestry as something that is relative, right? That all life made of DNA is relative, and our relatedness to one another is relative to scales of time and size. Humans, the variation that's in humans is negligible, right? Like we are so similar to trees and fungi relative to all the genetic diversity and all the species on the planet that when we talk about what's different within and among human groups, we're talking a really, 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 really tiny proportion of the genome. So I just think that perspective is important, that while we tend to really hyper-focus on our differences as humans, and that's mostly because we're visual creatures and we're, primitive mammals trying to make sense of our environment and determine what's a threat and what's not, that we make meaning out of those differences without really understanding how insignificant they really are in terms of who we are as a species. So what are those differences in the 0.1% of the genome that humans can possibly differ, and how do we get there? Well, it has to do with our migration patterns out of Africa, which is the original home of anatomically modern humans. We're all descended from sort of one mother population somewhere on the continent, and population geneticists quibble about whether that's in different parts of the continent, but let's just say for now it's somewhere around South Africa. And I like to use this analogy for those without a sort of a background understanding of genetics, like all of the genetic variants that existed in the original human population was like a big jar of jelly beans with all of the possible colors that you could possibly have. And you just sort of take a handful of those jelly beans or those genetic variants and you say, oh, this handful of jelly beans is going on the backs of these people who are going to march up out of Africa, and they're going to migrate and settle in this other area and then have population expansion. From there, there's a subset of those variants that travels to the next location, such that the further away you get from that home population where all the genetic diversity is present, you get less and less of that genetic diversity. And then you have population expansions, which is those bottlenecks, right? Where you have higher frequency of a fewer number of variants because it's a smaller subset of the original diversity that's found in humans. And so that's what we're getting, right? When we do European GWAS, we're pretty much just focusing on a very small subset of the variation that's available to contribute to health and disease. And that's all random, right? That's genetic drift, kind of random jelly bean grabbing alleles out, but that's not the only reason we see differences between populations. Obviously, there's natural selection over long periods of time. There's genetic adaptations to particular environmental conditions. So one example is this fatty acid enzyme variant in Greenlandic Inuit at 98% frequency in the population. Almost everyone has it, right? So this is thought to have helped the Inuit populations adapt to a diet that's high in animal fat and without giving them heart disease, right? But this variant also plays a big role in height in everybody. But the reason it was never found by GWAS done in height, of which there have been many in European populations, is that that particular variant is only 1.7% in the European population. So even though this is an important variant for height, it was only found in Greenlandic Inuit population because the frequency was high enough that we could actually detect it. So that's just important to consider, like what are the implications for research and a lack of diversity. Another example, and this is, I think, probably the most common among medical students and others who just take a little bit of genetics. Maybe this is your only population genetics lesson, right? That sickle cell anemia is an adaptation to the presence of malaria in a population, such that there's this advantage for people who are heterozygous to not who are not getting malaria, who are not getting as sick from malaria, but that same variant actually causes sickle cell anemia. And so oftentimes you'll see maps only of the continent of Africa where they're comparing, but I like this one because it shows you that parts of South Asia, the Middle East, Mediterranean, Southern Europe, all of these regions that are endemic with some form of malarial parasite also is endemic for sickle cell anemia. So this is not a Black disease, as many of us have been sort of taught or have assumed due to lack of nuance in our training. This is due to an environmental adaptation. And that happens to be at higher frequency in some populations than others, but that's all about the presence of malaria. It has absolutely nothing to do with race or racial identity, right? And similarly, lactose persistence or lactose tolerance, which is lactase persistence, or the ability to digest milk after a certain age, was an adaptation by cultures that relied heavily on livestock for survival in certain, maybe like a famine condition or something. But this is something that's evolved in many parts of the world. And yes, it is highly prevalent in Europe, probably because of those famines and need to rely on livestock, but it's also happened in other places. So when you have white supremacists chugging milk on YouTube to prove a point because of their superiority, what that does is it's a racialization of a trait that it has nothing to do with race and everything to do with whether you had to drink your livestock's milk to survive or your ancestors did, right? And that has nothing, again, to do with race or whiteness or whatever. So this constant issue of a lack of definitional clarity and separation of concepts between what's genetic, what's ancestry, and what sort of society and culture that's telling us about race and racial identity, and that those are separate, and that when we conflate those, we do a disservice to ourselves as a society, but also the accuracy of our research and our clinical practice. Because fundamentally, that the idea that there are real, meaningful biological differences by race that are due to genetics is based on not genetics, right? It's based on the desire to keep people oppressed, to have differences in allocation of resources such that white supremacy remains, right? So in the Jim Crow era, you have people being sent to different wings of the hospital, sent to different clinics. And the justification for that was that there needed to be Black medicine, right? There's white medicine and Black medicine. There's some fundamental difference on the basis of race that was used to justify the separation and the inequality and discrimination of the time. But we've known for a really long time that this isn't actually the case, that this is a myth, right? Designed to uphold white supremacy. What's real is that for decades, we've had population geneticists, Dick Lewontin, probably the most famous in the 70s, coming out actually doing the analyses, looking at genetic differences within and between these sort of traditional racial classifications or groupings, and finding again and again that the vast majority of variation you will find at the genetic level is within each of those groups, right? So you pick two random people from within a group, they're more likely to be different than two people that you randomly pick from between those two groups on average. So it's really important that we stay rooted in this knowledge that that in the 1970s you had Dick Lewington saying there's no justification to continue classifying people by race because it has no genetic or taxonomic significance. Okay, but in the US, for various reasons, we have this racial classification system that is baked into our system of government, right? So the US Census collects these categories. This year they have some more granularity. You can choose multiple. All of these are like recent innovations of the census that weren't always available. Now we have an other option, so people can other themselves, right? Rather than check one of these boxes or select multiple, again, recent. But these are not too dissimilar from those taxonomic groups you saw in the 1970s. Some different naming, some semantic differences, but fundamentally we're operating on the same basic system of racial classification. And what happened in 1993 is that Congress directed the NIH to collect information and report information on all of their study participants based on the US Census categories. The reason this came about was because actually the Women's Caucus and the Black Caucus basically were advocating for greater inclusion of women and minorities in biomedical research. So they're saying, hey, you need to track this, we need to see who's being included. This was an equity and a diversity, equity, and inclusion regulation, right? Saying that NIH needs to come up with a regulation that will help us track disparities in research participation over time. That was a laudable goal. The problem is that researchers then interpreted that requirement to report by those categories as the NIH telling them what their analytic groupings could be once they have diverse study participants, right? If you only had white people in your studies, you never had to think about this before, but now you're thinking about it and you have these neat categories that the US government is providing that you report study subjects, so why not include these in our research protocols as the analytic categories of interest, right? So what happens? These findings come out in the research, the categories are integrated into electronic health records if they weren't already from the Jim Crow era, right? How do I know where to send somebody in a hospital? You have to have their indication, their racial identity somewhere on their form, so you know kind of where they need to go perhaps. But in current day, we have electronic medical records, clinical forms, and all sorts of other data throughout the healthcare system that use this basic framing of check a box for all that your diversity is encompassed by. So this is an early study we did for the ClinGen Ancestry and Diversity Working Group looking at clinical lab requisition forms. And these are screenshots from 10 different forms from different labs. And I'm sorry if it's too small for you to see, but basically you have some with, very, very few categories. These are just basically census categories like minus Native Americans plus Ashkenazi Jewish or something, right? Plus Caucasian minus white, which I think we should all stop using the word Caucasian. We can talk about that in the Q&A if you're interested as to why. But in any case, there's a ton of granularity around the European categories. And then like on 9 out of the 10 forms, you have this wonderful Hispanic. What does that even mean? Why do you need that on a clinical genetic test requisition form? I don't know. So these are things that we're trying to test and investigate and like empirically look at from a research perspective. Why are we doing these things? Is it useful? If not, what's the how can we start to move the needle on all of this? Because it's just remained in the sort of unexamined, unconscious part of what we do, and we just take it for granted as what we've always done, and so what we need to continue doing. And I'm trying to break that down using science. So back to this sort of pipeline I mentioned before, we have this issue, right, of translating basic research into clinical genetic testing because we are not looking at all the variants that are possibly out there or that we know are out there contributing to health and disease. And then we have to make interpretations based on those tests. So there's heterogeneity in the data you have. So if you have all these different test requisition forms that are being used by different clinical labs, does that affect the interpretation? What if one form has all this granularity and another form doesn't have any information? Which lab has better outcomes in terms of their interpretations of their genetic findings? I don't know. So it's really unclear. So we wanted to learn more about this and actually look into this question of how race, ethnicity, and ancestry are used in research and medicine. So I want to tell you a little bit about what we did around that. But first I'll pause just to get a sense from you all what you're thinking about, or maybe in your own experience. Which of these measures of diversity do you think is most often used in biomedical research and medicine? So I'll give you just maybe 45 seconds or so to respond to this. On your poll everywhere, you can text. You should be able to text on the same line without texting again into the system. Okay, seeing a lot of responses around race. I'm looking up, by the way, because this is where my results are up there. Okay, quick question. Just anyone feel free to jump in. Since I don't think could you select multiple options, or was it just, if it was just one selection, how many of you who selected race would also have selected ethnicity, but not ancestry? Curious, or maybe you can show of hands on, you can do the hand raise function. Okay, a couple. All right, so that's good information to know that maybe you would have selected race and ethnicity, but not having that capability. That's helpful instead of ancestry, because you could say all of these, but people are mostly not saying that. So okay, mostly race. Good at race/ethnicity possibly. So thank you for that. I think you'll find the results pretty interesting of what I'm about to describe. So as I mentioned early on, we did some empirical things, some basic investigations into clinical labs and what they're doing, but really to get at the heart of what we wanted to know, we felt like we needed to design a survey and actually talk to people doing clinical genetics to understand what they're thinking about. So we designed survey questions both within ClinGen and with our partner consortium, the CSER project. And so after we developed questions, I actually did 11 cognitive interviews with folks, really pretty high level folks across all these different professional areas to make sure that the questions were getting at what we wanted to understand. We disseminated the survey through professional societies, through our consortia, and through our sort of social networks. And then I wrote a custom Python survey analysis tool to get some of the results. So this is published in the American Journal of Human Genetics. You can look it up. We got about 450 respondents, which was pretty great, and a good representation, I think, across the different clinical genetics professional roles. And then we also had a decent number of non-clinical researchers as well, I think mostly through the outreach through ASHG. So that was really great. And there may be some of you on here who participated in the survey. Thank you very much for that. So in general, and if you look at the paper, there's more detailed breakdown about kind of what terms resonated with people and thinking about race, ethnicity, and ancestry. So I'll let you do that on your own. I don't really have time to go through that today, but the main take-home message is that 76% of respondents were either somewhat confident or not at all confident in describing the similarities and differences among race, ethnicity, and ancestry. So that distinguishing between them and thinking about what kinds of information are they providing and for what purpose, that's not really, people are not solid in their ability to think through those similarities and differences, at least according to the results of our survey. And we also wanted to know specifically how important people felt different measures of diversity were in their work. So we have, different ways of asking this about different functions in clinical genetics. Again, if you look at the paper, there's more details. But overall, all three, race, ethnicity, and ancestry, were considered important, at least somewhat important, by the majority of researchers, or by the majority of people who are doing clinical interpretation of genetic variants. So these are people who've told us, yes, I work with patients, I interpret genetic test results, and this is what I think about these terms. So ancestry obviously actually gets a big spike here. So there are more people who felt like ancestry was either important or very important relative to race and ethnicity. And we were curious about this. Okay, well, if folks believe that ancestry is more important for clinical interpretation of genetic variants, how often are clinical labs actually calculating ancestry or providing ancestry results on their reports back to the clinicians so they can actually make their interpretations based on ancestry information? And we were actually pretty surprised to learn that almost never is this actually being provided, right? So, and the only people who said often or always to this question, how often do you calculate genetic ancestry from a patient's DNA for the purpose of clinical interpretation? And we asked this in different ways. Do you have it on the forms? Do you get the results? Like, it's the same across every way we asked it. But this was just, you know, good example. The people who are often or always, I can't tell who they are because it's anonymous and I wouldn't want to re-identify them, but I did look at sort of what are their, you know, institutions, if they provide their professional affiliations. And it looks like most of these are people who are involved in clinical research. So they're doing the ancestry analysis on their patients, possibly for clinical interpretation, but they're only doing that as part of a research project, right? Not because that's like become part of their typical practice. So it's like, all right, so all three, race, ethnicity, ancestry, are considered at least somewhat important for your work, with ancestry winning as the most important, except almost nobody has access to ancestry. So if information about a patient's race, ethnicity, or ancestry is used in any aspect of your clinical work, from where are these data obtained? Well, 94% told us that race and or ethnicity information is reported directly to them by the patient. So ding, 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 everyone in this seminar, many people in the seminar win who said you thought race or possibly race/ethnicity is primarily used. You're exactly right. That's pretty much what's going on. At least in clinical genetics. And then 18% admitted that race or ethnicity are recorded by another care provider, possibly without asking the patient directly. So this isn't even like self-identified race or ethnicity. This is a clinician, either someone who's doing an intake form or a nurse or your doctor coming in and just checking a box on the person's behalf or putting in the system, and possibly not even engaging that person in conversation about their background. That could be useful information potentially. So this is, you know, big problem, right? So you have this clinical interpretation issue where the data coming in, potentially garbage, data coming out, also potentially garbage, but useful probably for mostly, you know, people of European ancestry, where the algorithms don't have to be tweaked or anything because there's most representation from those populations, right? And at some point you have to give people a diagnosis. So you might have more uncertainty. In fact, we do have more uncertainty across populations, in non-European populations. And there are limitations in terms of diagnoses and treatments when you're factoring in something like race or ethnicity, but you're not able to actually give anyone better information as a result of it. It becomes a sort of exclusionary thing where it's like, well, because of your race or ethnicity, we can give you less information, or the information we have to give you is less accurate, as opposed to making it more useful. So what are the consequences of this? Well, there are many, but to summarize, three main ones that have been well documented in literature is that there are higher rates of variants of uncertain significance in individuals of non-European ancestry who get genetic tests and the doctors say, sorry, I don't know what to tell you. It's uncertain because we don't have enough data from people with your genomic background to make a determination. False positive diagnosis rates are elevated in African Americans compared to European Americans, and false negative diagnoses are more likely in individuals from populations that are not well represented in clinical databases. So what this means is that you're more likely to possibly have a genetic condition that can't be found because the genetics they're testing are from a different genomic background, right? So that would be those false negatives. And then false positives is where you have a link, a really tight link between the marker the genetic variant that you're looking at and some outcome of interest in a European population, but because of the different structure of linkage disequilibrium in different genomes, meaning that possibly in some generation, at some point in time, link between those two variants on the chromosome got broken up through recombination, and so then it's actually not tagging the causal variant in a different population, whereas it is in the Europeans. Why you might have a false positive that actually happened, and this paper talks about it. The Man Ray paper, so I encourage you to look at that. It's an example of hypertrophic cardiomyopathy and false positive rates actually impacting people and their treatment. So once we have these biases all baked into the pipeline, then all the evidence is curated, and the knowledge base, as I mentioned, then informs clinical care and drives future research. And overall, this is biased towards whites and people of European ancestry, and the genomic knowledge base is incomplete and imprecise. So where do we go from here? They're not defined. They're used all over the place. So how should diversity measures be used in research and medicine? This is the million-dollar question, right? Or probably more than that at this point in time, since the government and then research agencies seem to actually be really taking this up as an important question to be investigated. So we're working on it. I'm curious to know, this is a hyper oversimplified way of asking for your feedback on this, and I'd love to hear more in the Q&A session. But if you could just respond briefly, which of the following do you believe should be used in research and medicine, if at all? And it's hard because research and medicine, possibly different use cases. So, open to hearing a little bit of verbal feedback on that as well, if you'd like to discuss. Give folks about another 45 seconds to respond. Okay. So a lot of people saying ancestry, about half, 42% saying all, 8% saying none. I'm curious if we could, would someone who said ancestry or ethnicity, we got a couple, someone who said ancestry be willing to share why you think ancestry? You can either raise a hand or just start talking. I see Justice or Anina.
Yeah, I think that ancestry just provides a more accurate representation of what's going on in someone's genome. And so looking at the patient in the individual sense, according to what their parents may have had, what health conditions their parents or their grandparents may have had, may make it more effective, the treatment more effective, and how they target that the person's specific genome.
Okay, cool. So ancestry could be more accurate in terms of understanding someone's genomic background, for sure. Thank you for sharing, Nina. Thanks for chiming in. Looks like you're fighting the good fight there in clinic too. Appreciate you for that. And then Justice had also raised their hand. I don't know if you wanted to add something different.
That was Justice speaking before.
Oh, that was Justice before. Oh, sorry about that. I missed that. You're both on my screen. So thank you for that, Justice. And Nina, did you have anything to add? And you're the one who's showing us.
Yeah, I'm Ashkenazi Jewish, which is like extremely inbred, and I know, firsthand that can be a significant driver of a lot of, genetic-based diseases such as cancer. So I think to ignore it would be losing out on what could potentially be a valuable piece of data. And it can also be done in sort of an unbiased manner. Like you can assign someone's genome to a predicted ancestry in a way that you can't just by looking at them. So it seems to me to be both a little bit more objective and historically quite useful.
Sure. Yeah, absolutely. For those population isolates you're talking about with bottlenecks, really important to know whether someone has ancestry from one of those populations, because that is clinically actionable, right? And useful for sure. Thank you for that, Nina. Okay, and then I see Christine.
Yes, thank you. The question I put ancestry, but then I question myself, well, what if you don't know where your ancestor come from? Then which one would work? So that made me think, maybe we should try all of these, because if we have one for instead of another, or another, like directly the father or directly the mother, then at least you have something. But if we solely put it on ancestry, then how will you know if you don't have any records of that, with all the migrations and everything that have occurred? That's really what then threw me away afterwards, and threw me off, I should say. And that's when I stayed with my mouth open, and I went, good point. Let's see what will happen. Anyway, that's my question.
Thank you, Christine, for sharing. I totally agree with you. That's something to consider when we're thinking about alternatives to asking things like race and ethnicity. You could consider asking, right, about someone's ancestors. And I think that's pretty common practice for genetic counseling. When they're taking family history, it's like, oh, would you know where any of your grandparents are from? That's part of that process. And for sure, there's limitations there in terms of who knows who their ancestors are. And in this country, there's a particularly painful history around why certain people don't know their ancestry while others have. My ancestors, as a white woman, that's part of my white privilege, is knowing my heritage because my ancestors chose to come to this country. Whereas a lot of African Americans' ancestors did not choose. They were stolen here. Their stories were stolen from them. Their ancestry, their history is stolen from them. And so that's a really painful thing to be asked in any setting. Oh, where are your ancestors from? It's like, I don't know. And that's really not my fault, but there's something I can do about it. So I hear you. And I think that issue is very important. And I think runs even more deeply than just the scientific aspect of things. But when we can look at somebody's genetic ancestry and compare to reference panels and say, okay, well, it looks like there's ancestry from this region or that region. While we can potentially get more information about someone's genomic background on like sort of continental level ancestry, the lack of genetic diversity in basic research in our reference panels means that we're limited even in genetic ancestry by who's been included in the research thus far. How much granularity can you really get when all the direct-to-consumer companies are saying, oh, well, you have 30% sub-Saharan African? Great. That's like more than half an entire continent where all the genetic diversity is. So how is that meaningful, right? Or useful? So even with genetic ancestry, because that would be like, I think a lot of geneticists would be that their answer to your question, Christine, is, oh, well, we'll just look at the genetics and they don't need to know anything. But even what we can say about the genetics is limited by who's been included and excluded from research thus far. So yeah, absolutely. Lack of information is always going to be a limitation regardless of what we're looking at, unless we really make some big progress.
Alice, you've got an interesting comment in the chat here from Will Good, who writes, I thought that under a proper ethical framework, any of these categories could be useful depending on what the research is. I feel like while different races may not have very different genes, treatment by the medical system and lived experiences are definitely different, and those differences could inform research.
Yeah, thank you for that comment. Looks like Will, appreciate that. And so maybe this is a person who answered E, and that was going to be my next question for those who answered, all of these. What might be the reasons? And this is, I think, a really good argument for all of them being potentially useful, right? If you're wanting to track racial and ethnic health disparities, if you suddenly stop collecting racial and ethnic identity information that's that you can longitudinally look at from past records, then how do you know what progress you've been making? And they run into this issue in European countries where it's actually illegal to ask citizens or anyone their race or ethnicity. And I was in Norway for 6 months and it is not legal to do that. And they're like, oh, well, it doesn't matter because we're all equal and we all have healthcare and we all have education and like it's all fine. And that's great. And maybe that's true. Or maybe there are systemic inequalities by racial and ethnic identities, immigrant populations, et cetera, that are not being, that you just can't even look at the problem because it's even illegal to ascertain or look at those data. So I definitely agree that it's important to be thinking about how we track health inequities and disparities over time, and how we track progress towards mitigating those disparities in one sense. And then I think in terms of genetic ancestry, there are certainly uses in research and in medical settings. So it's just a matter of figuring out what's the appropriate use of the appropriate category, and can that be justified scientifically and ethically? And that's really the goal. Sounds great, really difficult to implement in practice, right? And figure out what those are. So we're working on it, but I appreciate that comment. I wonder too, if anyone who said none of these would be willing to share their perspective, since we just talked about the opposite. I see Joel's hand, actual hand up as opposed to a virtual hand. Feel free to share. Yeah.
I didn't clear. Who are you identifying?
You.
Oh, I'm sorry. Yes. I hope you'll talk about the negative consequences of collecting the ancestry or racial information. Yes, there are some advantages. The norm tests that people use by race may have some advantage. But overall, it provides grounds for discrimination that'd be much worse than whatever we gain by doing it. And it may be that in the end, the argument against collecting the information may be compelling. Will you talk about that, please?
Looks like I was muted. So thank you for sharing that in the chat, and also for sharing verbally. I'm going to turn it back around to the audience, and also maybe to you, Joel, since you have thought about this. You know, what do you think the potential consequences are? Like, what's that balance, right? Of what's the risk and benefit of not collecting any of this information versus collecting it? What's at stake in both directions, do you think?
I think the stake is very high. I think we have good examples of use of that information for negative consequences. Quick example, the chief of police in Los Angeles some years ago used the argument that that police in Los Angeles had to use a particularly strong form of the chokehold on Africans because they had a different physiology. And therefore, the use of racial information became a rationale for discrimination. My reading is that overall, that's most common. And therefore, we would be ultimately, those who are disadvantaged would be ultimately better off by not asking those questions.
I see. So the point is that if we can move beyond classifying individuals in this way and thinking about people as belonging inherently to different groupings, then that will sort of inherently allow us or enable us to move beyond discrimination and sort of discriminatory policies on the basis of that. Did I get that?
Yes, that's the direction.
I see.
And that, I mean, think about advisors who tell girls they can't do science because they're girls. They might provide very different advice if they didn't know the gender of the student they were talking to.
Yeah, that's true. And I wonder how we might construct a society where you wouldn't know the race of a person by looking at them, right? Is that ever something we could live, right? Like, I think so often, and I'm not disagreeing with you, Joel, I'm just offering, you know, an alternative perspective that perhaps we focus, that perhaps the concept of race and the concept of these categories and who belongs in what category, that's all a distraction from systemic racism and inequality, right? Like when we talk about race as something separate from racism, then we're giving it power to be something meaningful, when really what it is a mechanism of racism or a mechanism of oppression and designed to maintain white supremacy. So I wonder if perhaps we could, if that would achieve what you're talking about, if we could still focus, you know, if we can still use the categories to track disparities, but then be really explicit that that's what it's for, that it's a proxy to measure systemic racism and inequality, as opposed to something that can be used to justify literally anything about biology or policy or otherwise, unless it's to lift up those communities, right? Possibly, just an idea. Okay, I see another hand, Christine, and then I can just finish my slides and then we can sort of open it up for general Q&A. Does that work, Jasmine? I see. Okay, cool.
Yes, I just wanted to pick up where the last person just talked about the issue of race. I think that it depends on the issue of power. What is that used for? For exerting a certain influence, or shunning a certain group away, or manipulating a group. So it's, again, what is the intent for doing such a thing? And maybe there needs to be a section in the research itself that explains this research is not for any of the, and then discuss or summarize the idea of exclusion or discrimination or certain tactics. So basically all I'm saying is that it has to be devoid of any connection to systemic racism. That's all I'm saying.
Okay, thank you for sharing that. Okay, so I'm going to just summarize what I've shared with you so far, and then we'll open it up. So what I'd like, if you were busy at the first part of this and your head swimming with all of this confusing, very deep concepts, what I'd love for you to take home is just what's on this slide, that there's no genetic basis for racial categorization of humans. That is well established. There's no global standard for the definition, collection, or use of diversity measures in genomics research and clinical genetics. Folks are working on it in lots of different ways and different capacities, but there's no consensus right now. There are biases in the research to precision medicine pipeline that lead to real health disparities in clinical genetics, and guidance is desperately needed on the use of diversity measures in both research and medicine. And there are a lot of really smart people working on this, a lot of them way before my time. I want to acknowledge all of the folks on the ClinGen Ancestry and Diversity Working Group, the CSER working groups who are part of this, our program officers at NHGRI, all of the other folks who were involved in dissemination and creation of the survey, some mentees of mine who've worked on various aspects of these issues. And I think probably most importantly, all the scholars and investigators whose prior work on this have made this project possible. There have been social science researchers, I will say most of them that have heavily influenced me are women of color. And if there's one book you read on this topic, I'd highly recommend Fatal Invention by Dorothy Roberts. There are a lot of people whose work on this have sort of laid the foundation for this conversation. So I'm just really grateful to have landed in the right place at the right time to be able to use the privilege that I have to speak on these issues and hopefully work with you all and others to make progress on this topic. So thanks for that. And I have some this open poll everywhere question that you can write some questions. I think maybe this one you can't text because you should be able to upvote and downvote questions that other people ask. So if you go to that link there at the top, you can type your questions. Also, I think we'll be monitoring the chat, but you can type them here and then upvote and downvote other people's questions. So thanks again for having me. And if you have to log off, feel free to email me anytime. It's apopejoy@stanford.edu. You can find me in the directory if you want to get involved or continue the conversations, but happy to continue with our Q&A.
Thank you so much, Dr. Popejoy. Oh my goodness, this was so rich. And thank you to all those who are already contributing to the discussion, whether just answering the polls or lending your voice to the conversation. While we wait for some of the questions to come in, I would love to pose a question from one of our students to you, if that's all right. So the student asks, why is Middle Eastern and North African identity classified as, or often classified as white under census or common survey forms? Is the Middle East / North Africa geographic itself too broad a category to classify diverse communities of people? And how does this then, how does the existence of this specific category then impact research or healthcare? So if you wouldn't mind taking it away, thank you.
Sure, thanks for that. That's a great question. I don't know that I can, answer it to full satisfaction, because I think there are a lot of reasons why that's problematic. I would say probably the reason why Middle Eastern and North African is considered like one category is because there is a lot of gene flow just around those bodies of water. And so if you look genetically at, sort of global continental level ancestry groupings, you might see folks from that part of the world like clustering close-ish to one another, right? So that could be part of the reason why they're grouped together. And then the fact that they're considered white, I don't know that I guess I've not come across that as a rule. There are folks who are Middle Eastern and from North Africa who identify as white, who have lighter skin pigmentation than other parts of the African continent, than other parts of South Asia or the Middle East. It could even harken back to, if you are seeing sort of some blanket categorization, like these are white populations, that could honestly go back to the invention of the word Caucasian, right? That being from the Caucasus Mountains meant something, right? That was something about like the Aryan race, which becomes the white race. But when you look at the people who are from the Caucasus Mountains, like in the US, they wouldn't be considered white, right? Because these are not fixed characteristics. These aren't just like objectively truthful traits, right? Like they're relative to the culture and the context and the time. At one point in time, Irish people were considered Black in the United States, right? And like in South Africa, in apartheid, Indians with really dark skin pigmentation could be considered white because they were wealthy, right? Like there's just, it's all relative, or you could sort of like, buy your way into whiteness so you could get certain rights and privileges, right? Like there's no there. So the idea that some people might say, oh, people from the Middle East are white, maybe that's just because the idea is that Caucasian means something more than what it is, which is actually sort of the last remaining vestige of Linnaean taxonomic categorizations of like sub-races of humans, right? Where subspecies, where you have the Negroids and the Mongoloids and the Caucasoids, right? We cling to this word Caucasian because it sounds really scientific, but actually it's just archaic, and it's something we inherited from basically a very racist classification structure. So I would argue that when we're talking about whiteness, we should talk about the racial category as is constructed to reinforce white supremacy, and not sugarcoat it with words like Caucasian that make it sound more scientific than it is.
I'll jump
I don't
in
know if
with.
that answered your student's question. They can chime in maybe if the student would like to, or share.
I'll jump in with a comment, and you don't necessarily have to follow this up, but then we have one question in your poll everywhere that you might want to look at. And my comment would be, there's a history to whiteness too, as there is to everything else. And of course, in the 19th century, when there were still waves of immigration from Europe, Italians and Irish and Polish people in particular were stigmatized, even though they were white. And the collective, the use of white as kind of a collective category that catches all of those immigration statuses is historically more recent. It's not the same, I'm not saying that they were identified racially in the same way that black people were identified during that time, but there were stigmas related to ethnicity then that are mostly collapsed now into the category of whiteness. So maybe you might want to respond to that, or you can go on to the question that's in your poll everywhere.
I actually think that comment really dovetails nicely into a discussion of this very provocative and wonderful question in the poll everywhere. Do I see my work in genetics in conversation with debates around modern articulations of eugenics? If so, how do you navigate those concerns? Well, originally eugenics, I highly recommend this documentary called The Eugenics Crusade. I think it's an American experience, The Eugenics Crusade. It's on PBS. I think it's freely streaming still. You might want to check on it, or at least you can get it through Canopy or Stanford. And it talks about the history of eugenics as primarily a way of promoting births among Northern Western Europeans. It was a way of sort of othering those white ethnic groups, as they were referred to at the time. And the reason that they focused on these white ethnic populations in terms of classifying them by feeble-mindedness, criminality, right? Assigning all of these very complex behavioral traits to genetics and saying this family has no more right to reproduce, so we should just sterilize everybody because they're all going to be criminals, was a way of doing ethnic cleansing within the white population. And the motivation for that, and the reason why eugenics was so focused on white ethnic groups, was actually because they felt like there were enough laws already on the books that kept people of color at bay. Like, oh, they already have less rights, less access. Like, that's not a thing we need to worry about because that's already taken care of. So now let's focus on these sort of white ethnic cleansing policies. That's not to say that absolutely horrific things didn't happen to communities of color during the eugenics movement. I would say probably disproportionately so. Those horrors were certainly applied across the board. And I think in modern articulations of eugenics, as someone mentioned, right, there have been forced sterilizations on imprisoned Latino women in California in 2010, right? So there are very real eugenics practices and policies happening today as a result of the inheritance of our culture of eugenics and really the founding of modern human genetics research by Charles Davenport, really, who was mentored by Francis Galton in the UK, right, to really come and classify people. And that's their whole thing, is classifying people. And how do we break up people into different groups and classify them and make that mean something and then assign it to genetics, right? This is genetic essentialism, it's reductionism, it's unscientific, it's biological racism. And because genetics as a field has yet to reconcile that history of the field, those concepts are free to permeate the culture of research and the society's culture unchecked because it's all unconscious. We're not addressing that consciously. So how do I deal with this? How do I navigate this in my work? Step one is, like, I taught human genetics for UC Berkeley last summer. And in my curriculum, we do an entire module on eugenics. Everyone's required to watch the Eugenics Crusade. We talk about it. We say, how can you learn human genetics or say you know anything about human genetics if you don't know the history of the field and how we got here and sort of what that means? So one is just like educating people about the history, talking about how what we're doing today, you know, can be connected to that. I think I can probably do more of that in my talks, you know, do more of talking about eugenics. Like, I still have a lot to learn as well. And, you know, and trying to figure out, right, what's the best way to navigate this space when there's so much at stake. But I think for me, it's all about not letting us off the hook as a scientific and a medical research community. Like, oh, that was in the past. No, we used to do that, but it was pseudoscience. No, that was cutting edge science at the time. That's what was considered to be the norm. And then the most horrific thing that I actually learned from watching that documentary was that not only did eugenics sort of become like othered as a pseudoscience after World War II, or when Hitler went running off with American eugenics science and ideals to justify his agenda. Then the Americans were like, oh, we want to separate ourselves from that. We don't want to have anything to do with that. But and at the time, the scientists were sort of withdrawing their support, right? You were seeing Thomas Hunt Morgan, the fly guy, who was like, I can't figure out the genetics of banding patterns on flies. Y'all are talking about genetics of criminality? I don't think so. Like, I'm going to quietly bow out of this. And there were other scientists doing the same. But then what happened after it lost scientific credibility is that eugenics policies, sterilization policies, forced sterilization policies were appearing on the books of states all across the country. So you have 30 plus states implementing eugenics policies after the science has already lost its internal credibility. So there's a disconnect here between what's happening in science, what we know based on science, and then what's happening in the policy space. So that's another thing, that the bridge that needs to be built is to make sure that the policies are informed by the science, and that the science doesn't pretend to be devoid of bias because somehow we're operating in a vacuum and not within in a society, within a culture. We have to acknowledge that, and we have to teach it, and we have to repair the harms of the past in order to move forward in an honest and earnest way, I think.
Well, thank you for that honesty. And I would like to now call on Ocheze Amuzi, who has a question for you.
Oh, hi. I hope you can see me and everything.
Yes.
I'm connected to my phone, but yeah, no, thank you. I've learned so much. It's been super informative, and I had sort of some questions based off of what Joel was talking about when he brought up his examples of teacher and the doctor and the cop, kind of just observing racial information and kind of using that to make decisions in the moment about the care they provide to community members and stuff. And just that action of just them perceiving is enough for them to adjust their behaviors and things like that. And I was thinking of okay, well, if just perceiving is enough to kind of change the outcome, what are the situations in which a doctor or teacher or cop or whatever isn't going to be able to perceive the person they're working with? And obviously telehealth is one of the first things that came into my mind. And I was just wondering if there was precedent or examples from research or whatever that you heard or that you know of race blind care where the provider can't perceive that information readily and maybe isn't provided, having that be a sort of pathway to more ethical and equitable care, if there are those benefits and what they've been in the past.
Yeah, so if this is a question for me, I don't know of any studies. If others do, I'd love for you to chime in of just really some strict, almost like a randomized trial, if you will, of sort of race blind implementation versus race conscious implementation. I'm sure there are people working on that. And we're actually thinking about different ways we can test this within ClinGen and with our links to clinical labs. I will say that for Stanford clinical labs, there is no question about race, ethnicity, or ancestry on the requisition forms that we looked at from Stanford labs. That could be because it's an internally insured health program, and 14 or 15 of the congressionally approved CPT codes for reimbursement health insurance are based on race, ethnicity. So it could be that you actually have to tackle the insurance reimbursement system and those classification codes that they use in order to even feasibly propose that we take these things away, because that's how a lot of genetic tests get covered. Throw a bunch of race ethnicity codes and hope that it sticks, right? So that's one hypothesis that we have, that has to be looked at. And is it possible that in Stanford, the clinicians are looking up the race or ethnicity of the patient to figure out, their clinical interpretation? Maybe, or it could be that they're offering clinical interpretations without looking at that information at all, because it's not on the requisition forms. And the only reason it's on other requisition forms is because of insurance or, inertia. So it's a good question. And I think it's worth, considering. And if other researchers have the ability and the capacity and the interest to take up potential studies to learn about what happens when you take that awareness away, I think that's really critical to coming up with the next steps. So thank you.
Jayla, would you like to ask your question?
Sure. Hello, thank you so much for sharing with us today. I learned a lot, and I quickly wanted to ask the question. So I've basically seen a really interesting study about the racial differences in bone strength and bone density. I think it was done specifically on South African women who were Black and white racially. So I wanted to know whether these studies are ever actually useful in not aligning with eugenicist notions, and is it important to do these types of studies, and maybe what we gain and lose from them? Yeah.
Yeah, thank you so much for that, that question. I think it encompasses so many of the issues that come up in this space all in one question. So I appreciate it. And it's, there's many layers to the answer. I hope I can do it justice. The first that comes to mind is the absolute frustration that I have with our tendency when we're grouping people into populations to assign a population mean to every individual in that group. So it's the overlapping bell curves, right? You've got one population, is whiteboard here. You have one population that or group, however you group people together, right? And this is their sort of distribution of some trait. And then you have another population that, looks like this, right? And so you have these two like population means that differ by some amount, right? And so that's the overall average difference across everyone who's grouped as A and B. But when you just do that and you're not looking at other metrics of uncertainty around those estimates, how useful is this really? Because it could be that it is the case, first of all, that all of this is the same. If you fall anywhere in the normal distribution of the overlap of these two curves, it literally doesn't matter whether you are grouped into this curve or this curve because it's all overlapping. So there are tons of people, right, in this curve who are over on this side and people this curve who are over on this side, and that won't change a thing, except we pretend that everyone in this curve lives here and everyone in this other curve lives here. It doesn't make sense. It's unscientific. And we do it all over the place in medicine when we're saying, oh, this group has this average bone density. Sure, but like, did you look at how widespread the distribution is, how much overlap there is? If you grouped people randomly according to like how many sips of water they had that day, are you seeing a statistically significant difference? Are you looking at other ways of grouping? Like, there's so many scientific problems with this issue, right? So that's one. The other is that it's something like bone density, right? Or like, intelligence as the Murray, you can use this if people are like busting out the Charles Murray bell curve of IQ and say, well, most people live in the middle, right? So that's step one to debunking that argument. But step two is saying this is a complex trait, right? Like studies have shown if you do lots of high impact, jumping or sports, running, you have increased bone density. And that's true whether you take up running or lots of jumping activities or even dancing, right? Like in older people. So that you can boost your bone density by doing high impact sports, in 65 and older populations. So the idea that something like bone density could be attributed to something like differences between Black and white individuals is just crazy, right? Because sure, there might be a genetic component, but there's also tons of environmental components. There's gene-environment interactions, there's gene-gene interactions. It's a super complex trait. And your starting point, which is the comparison of these two categories, is meaningless because it's a social cultural framing, and you're trying to make some biological meaning out of it that, sure, if you observe it, you can say is true because you've observed it, but what assumptions went into the framing of that study in the first place? And so when you ask me the question, like, can this be useful? again, I'll go back to, if we are doing a study that you think people might be harmed unless you do this study, and you need to make sure that you're not harming people because of real differences, whether it's environmental or genetic, doesn't matter. If you're doing some intervention that's going to hurt people because of their situation or their biology, whether that's environmental or genetic, do the study to make sure that you're not hurting people. But if the point is to like change the threshold for diagnosis for like preventing people or excluding people from treatment because you say, oh, well, it's okay if they have a little bit of loss of bone density because overall that people from this group has a higher bone density at baseline, that's not a justifiable reason to do the study to begin with, or to make any implementation, clinical implementation that's based on that population level difference in means, right? That's unethical, not let alone unscientific. So why are you doing this study? Why are you grouping people the way you're grouping them? What motivated your decision? How is it justified scientifically, ethically? And then like, what is the implication? And how are you going to use the results of this study if it's going to potentially harm people? If you find some association, then maybe don't do it, right? I hope that answers your question. It was yeah.
Yes, thank you. I thought that was a super thorough answer. I really appreciate it.
Okay, good.
Thanks.
Okay, folks, we're out of time. Thanks for coming today, and hope to see you next week for Professor Keith Whaley from Princeton on pain. Alice, you'll stay on a minute. We'll debrief